the oscillations are really different. Also, the shape of the curve, if you sort of smooth out the peaks, the shapes of the curves are very different, right? So particle size has a really big impact, particularly in the forward direction on the scattering. Okay. It's because it's been modeled with a single particle that's a sphere, and so you're essentially looking at the emission at various angles where there might be constructive or some destructive interference, and so you'll have bright and dark patterns and that internal reflection of that particle. Okay. Okay, so then we're talking about size distribution. So you think about things that are very small versus very large, and we look at the distribution from, say, viruses to colloids to phytoplankton to nanoplankton to microplankton like diatoms. And if you measured the size distribution in the ocean, you can characterize it by the slope of this line. So if you have a steeper slope, you would say that you have more small particles in your sample. If you had a flatter slope of your size distribution, you would have more larger particles, right? So they'll be talking about this on Wednesday, about how to characterize this size slope. So we looked at now, instead of considering one particle, considering a population of particles that we modeled based upon whether the slope was flat or steep. Okay, so flat or steep. So the yellow is dominated by more small particles. The red is dominated by more large particles. So what do you see in the backward direction? Larger particles scatter less in the backward direction, and this is normalized by the total. So we have an equivalent volume of material. We've just partitioned it into different sizes. And what do you see in the forward direction? So this is on a linear scale. I've blown this up. So large particles have less backscattering, but more forward scatter. It reverses. So they have a steeper volume scattering function than do smaller particles. Right? So in a relative sense, there's relatively more backscattering per total particle volume by smaller particles, and relatively more forward scattering by larger particles. So if you think about the ocean, where you have dominance by small particles versus where you have dominance by large particles, they're going to have very different forward to back scattering ratios. Okay. And this is the same exercise, but changing the index of refraction for one of those size distributions. So again, you have much higher backscattering. This is normalized per the total particle volume. Much higher backscattering by the refractive particles. And in the forward direction, it's not very different. So let's think about that. You have size distribution of particles. You allow them to all be very refractive or not as refractive. The biggest difference between the index of refraction for a population of particles is in the backward direction, but not in the forward direction. So what dominates forward scattering? Which process? Diffraction. So diffraction is relatively insensitive to the index of refraction, but it's very sensitive to the size. And so you can make use of that by measuring diffraction that can provide you information about the size regardless of the composition of the particles. So this is why the list is a size meter, not a particle composition meter. It measures diffraction. It doesn't matter if you have a 10 micron sediment or a 10 micron phytoplankton, it will invert its signal and tell you that it's 10 microns, which is good, because if it were sensitive to size and composition, then you couldn't pull out information about either. So diffraction provides you with information about size.
I'm confused about this.
So it's a population of particles, and I've taken the same population. They've got some size distribution from microns to tens of microns. And in one sense, I've said, okay, I'm going to have every one of those particles, even though they're all different sizes, I'm going to say they have an index of refraction of 1.22, which means that when light goes through those particles, it slows down a lot. Okay? I'm going to contrast that with some particles that have really low index of refraction, but the same size distribution. And because it's a distribution of different sizes, that's why we don't see all of the oscillations in the volume scattering function. It's nice and smoothed out. Because I have the same size distribution and the same concentration of particles, I can, in this case it doesn't matter because they're the exact same, but I've basically normalized it to the total particle volume in the water, so that we're ignoring any differences in volume, particle volume per volume of water. And so what you see is that in the backward direction, the volume scattering function of the highly refractive population is much higher than the backscattering of the not as refractive population. So backscattering is very sensitive to composition. Composition, which determines the index of refraction. If it's a phytoplankton, it would look something like this. If it's a mineral particle, it will look something like this. So for comparable particle volume, you will get a lot more backscattering by minerals than by phytoplankton. So an ocean full of phytoplankton, not very bright. An ocean full of mineral particles, very bright, because it's the backward direction that is getting the downward light scattered back to your eye. Yeah.
How do you control for the index of refraction?
I set it. It's a model. You'll get to do this in a couple weeks. But the forward direction is not sensitive to composition. It's sensitive to size. And if I go back to, this was the differences in size. These had the same index of refraction, but these are dominated by big particles. This is dominated by small particles. Same total volume of material, but packaged in different sizes. Bigger particles versus smaller particles. So diffraction, very sensitive to size. Very insensitive to composition. Yes?
So if you increase the index of refraction, like what I see from this graph is that you have more scattering for all direction. It would be like more similar. So like you will.
So overall you have more scattering by highly refractive particles.
Yeah,
Yes.
more scattering.
Yes. But not in the first couple of degrees. This is on a log scale. This is on a linear scale. So in the first couple of degrees, they come together.
Why do you have more scattering like in total?
Because the total scattering, how do I calculate the total scattering for this population versus this population? How am I going to calculate a scattering coefficient from the volume scattering function? You have to integrate under the curve. So, yes, in the first 2 degrees, it's not they're the same. But when I integrate over the whole, you will have a higher scattering coefficient for the refractive particles compared to the non-refractive particles. But at specific angles, that can be very different, and it's very different in the far forward. So when people design instruments, they look for things like this that you can exploit. And so if you find that a measurement in the far forward direction is insensitive to index of refraction, composition, but very sensitive to size, there is your opportunity to design a particle size analyzer. If you have a backscattering sensor, you can see that your backscattering is going to be very sensitive to the composition. Less sensitive to size, but still somewhat sensitive to size. So you might think about how I would maybe measure the whole scattering, or the backscattering divided by the whole scattering, and maybe that would be a proxy for something. Or maybe I would measure a couple of different angles and find where they come together and they spread apart, and then maybe be able to separate that and get more information. So by doing these sorts of models, while they're not perfect for the ocean, particles in the ocean, they begin to give you some insight into how to exploit these signatures into useful information about the materials in the water. And so this is the optical proxy approach. We're going to measure diffraction and get size distribution. Okay, and there's a lot of assumptions in the inversion, but it's making use of the lack of dependence of diffraction on index of refraction. Mm-hmm.
What's the axis? What is
This is from 0 to 2 degrees. It's this part right here. And this is log scale. So you see very, very big difference. This is linear scale. So even on a linear scale, there's not differences. Just in the first 2 degrees. So it all splits apart right here. So you could imagine having a sensor that maybe measured, 1 to 2 degrees and then maybe 20 degrees, and that would allow you to see very, very big differences. Okay. So this is from a paper that Emmanuel wrote a couple of years ago that I really like, where he looks at making measurements with backscattering sensors or scattering sensors. And if you make your measurements of scattering or backscattering and you divide by the total particles in the sample that contributed to that scattering, so again, you're keeping that constant. You're not allowing for changes in total volume of particles. You can look at which size classes of particles contribute most to that signal. So this is backscattering. So you're looking at the backward part of the volume scattering function. And you can model that backward part for particles of different sizes and different indices of refraction. And so very refractive particles, not so refractive particles. But where in the size distribution do you see the maximum backscattering? So here's the scale down here. So these are 0.01 millimeter particles, or these are microns. Or these must be microns. Yeah, okay. 0.01, do they contribute a lot to backscattering? Nope. Do the 0.1 micron particles contribute a lot to backscattering? Starting to. There's 0.02. What about the 10 micron particles? Do they contribute a lot? No. So if you take a certain particle volume and you divide it into 10 micron particles versus you take that same particle volume and you divide it into 0.5 micron particles, you're going to get a ton more backscattering from those small particles than the large ones. Okay. Here's the same thing with scattering coefficient. So, yeah.
Why do you have this minimum?
Why do you have this minimum?
No, like you have the maximum of the backscattering, and then you have kind of the minimum.
This one is a different index of refraction.
No, the, like
The
deep.
yeah.
It's Emmanuel's modeling.
I mean, this is mean theory output. So you assume homogeneous spheres. For each side, you do a calculation for a single value, so it might be that it's I don't know. It's a good question.
But I think what you can take from this is that small particles dominate the backscattering, and larger particles dominate the total scattering coefficients. So if you have side-by-side measurements of backscattering and total scattering, you're sensitive to different constituents of the particles.
But this has been challenged now, by Collin as well. Because when you remove these assumptions about homogeneous spheres, things do not necessarily behave like this diagram. The deeps you see gets much more smooth if there is
Smoothed out.
So,
Mm-hmm.
if these were either weakly or non-absorbing.
But we did an experiment where we took a big suite of particles and we measured the backscattering and the scattering, and then we size fractioned the particles and we looked at the contribution, and we found that it was the smaller particles that contribute. Yes, we did.
But we found a big particle contributed much more than we predicted.
Much more than me predicted, but that the backscattering was dominated by the smaller fractions, and the bigger particles were dominating the total scattering. But not according to, not the same dependence of me theory, but it is the smaller particles. That could be a project. Okay. So consider what information scattering can provide you, and that will tell you what you want to measure. Are you interested in the total particle load? Are you interested in maybe the diffraction part of forward scattering? Is it the back scattering? Are you interested in the entire volume scattering function and the information you can get to that? Obviously, if you measure the volume scattering function, you can derive all the other things from it. So doing this is the best thing to measure, because then you can derive everything from it. Okay. And so scattering closure is where you make all these measurements and maybe match it to models, or you measure the total scattering and then you try to reconstruct the total scattering from the scattering properties by all the particles. And this is the reductionist view that Darius and Kurt Mobley did a number of years ago, where they actually looked at the volume scattering functions for a variety of different particles in the ocean, and then they figured out how many of each of these particles you might find in the ocean, and they added it all up together and tried to reconstruct the volume scattering function that had been measured in the ocean. So this is the volume scattering function for, I think, off of San Diego Harbor in the dotted line, and this is the reconstruction based upon the sum of the scattering properties of individual particles all added up together for a typical ocean. So there's some nice closure.
You notice the reconstructed phase function, the solid line there, is less at the backscatter directions than the measured one. And there's been this decades-long thing about what's missing in our understanding of the ocean that gives us the high backscatter we measure, because we add all these particles up and we throw in colloids and we throw in viruses, and doesn't matter what we throw in, we still can't predict enough backscatter. And there was once upon a time people thought, oh, it's viruses, because they just discovered there's a gazillion viruses in the ocean. But then the viruses are there, there's like 10 to the 12th per cc or something, but they have such small scattering efficiencies that they just don't matter, at least according to the calculations. Same thing with colloids. You know, there's a lot of them there, but they're so small, and optical efficiency per particle is just not enough, that even though there's a lot of particles, that would explain it. So people are still trying to figure out why backscattering in the ocean is as big as it is.
Yeah. People think of bubbles and foam and
Yes,
things that you can't put in a lab.
So there's bubble and foams, there's the phytoplankton internal structure and shape would cause more backscattering than you predict from this theory. And it turns out when you run them in lab, indeed the values are significantly higher than other people say, but is it the arsenic, is it the bacteria that are in there that are scattering? And lately there's a whole body of work by Zhang where he needs small particles to explain the VSF that he measures. Nobody measured those particles, but his inversion.
So it's this.
And that's basically like saying, okay, there's a difference here. I don't have enough things. It must be small particles.
Exactly. It's exactly that.
As opposed to saying that maybe your instrument isn't measuring well in the back. It could be that also, right?
Well,
Yeah.
I mean, they see, you can radiate transfer this, how much will the reflectance?
Yeah, that's how, I mean, it started with Morel and Hahn and others, that it just, we measure more light coming out.
Then we should.
We can have that.
Yeah. Active area of research. Questions to be answered.
Backscattering, we go on since like 90
Exactly.
Has anyone checked these at different depth?
What do you mean, the volume scattering function?
Yeah, whether the prediction or total backscattering fits the observation at different depths.
Emmanuel?
I don't know what you mean by testing.
Validating?
Well, the idea is that you cannot account for the total backscattering that you're measuring. Right?
So you're inferring from remote sensing.
So that's my question.
So
If you go different depths, then the bubbles will have a different contribution, and that's.
Yeah, people like Stramsky did a very clever experiment where he had a pressure tank. And so it's at atmospheric pressure, there's presumably lots of little micro bubbles, so he measures the scattering. Then he pressurizes it, which would collapse the little micro bubbles down to nothing, but the instrument's still inside, so he measures scattering again. Didn't make any difference. So he concluded that he couldn't explain it through micro bubbles, because even if it's like 100 atmospheres or something, there should be no bubbles there, and it didn't really change the scattering. So yeah, there's a lot of people that look at things like that. And then, you know, it now gets into the point like, well, it's things like particle shape and internal structure, because non-spherical particles generally have more backscattering than spherical particles for the same volume and index refraction and all of that. So then, you know, it becomes so hard to do calculations for non-spherical particles that it's hard to sort of, you know, have an equivalent of B theory that will work for these fractal shaped particles or whatever. And so that's kind of the current state of the art, is trying to do those sorts of calculations. But
And so the question is, does it account for all of this, Kurt?
So some of
Right.
the techniques that Collin talked to you about yesterday, where you do successive filtration with different size filters, you can do on cruisers, and Georgia has done this while measuring backscattering in a box, and he accounts for the scattering from the box size pretty carefully. And what he finds with those is that he gets more backscattering from the large fraction that could be predicted through the cruise track.
Normalized to the particle volume?
In proportion.
Proportion,
In what
yeah.
you'd expect them
Yeah.
to provide.
Yeah.
Relative to the. Again, so the explanation is because phytoplankton that dominate this larger fraction have internal structure, and he believes that it's internal structure. I think shaped, or whatever. Anyways, the peculiarities of the phytoplankton make them scatter more than you would predict from the.
When you did the experiment where you looked at aggregated and disaggregated particles with the list,
Yeah.
did you look at the total scattering,
I mean, we have the total scattering from the list, but not backscattering.
but not backscattering?
That's an interesting issue.
Yes, it is.
Like change-form organisms look like that.
So, yeah, so you would model them based upon their individual cells of the chain, likely, right? But they're not independent because they're in a chain. And, sometimes the chains are not chains like this. Sometimes they're helices, right? And so the way that I think about them, when I look at them under the microscope and you can see the scattered light off of them, that's what it's doing in the ocean. So like when you do microscopy and you see sort of scattering coming around the spines of a diatom, that's what it's doing in the ocean too. So the spine is going to have a scattering, volume scattering function, and the cells, the chain of the cells, and then it depends on how tightly coiled it is. It depends on what orientation, if it's helical this way versus if it's helical this way. So that's not an answer, but
Nobody checked that.
I think you could.
You could do culture. You could do culture
Yeah.
experiments.
Hmm. But you could do culture experiments in a flow field where they align, right? Where you get particles lining up in a flow field and look at the scattering, backscattering at different angles relative to the flow field, and I think it would be different. So people did look at, Darius Bogutsky looked at scattering off of turbulent structures. I don't know where that ever ended up happening with that. It was the small angle, yeah, it was all the forward scattering. Yeah.
That's small changes in initial refraction. As you know, turbulence in the ocean, there's slight thermal and salinity changes between the eddies and the levees, and those will cause the direction of light. But in very small forward angle, it's likely scattered from a large particle.
Mm-hmm. You can simulate that in the spectrophotometer in a long path cuvette. If you have a little bit of fresh water in there and you pour in salt water, you can turn it on its end and look at it, and you can see the turbulence of the mixing of the salt and the fresh water. And the fact that you can see it by eye is because it's scattering. And so you can actually visualize the scattering off of turbulence. So,
And it's an issue with the least. If you go
yeah,
with the least,
huge.
from fresh water to salt water, you actually get shearing effect. The
Shearing.
direction of light affects the water.
Mhm.
So something to keep in mind. When you look at very near forward scattering, that even homogeneity's intensity in the water will really about people.
Yeah. I'm done.
And you see
I'll show you a pretty picture at the
swim in
end.
the ocean in the place where fresh water comes in, you can see it in your mass. It's pretty cool.
Yeah. Yeah, if you go through the gradient. Yeah. So, the importance of scattering in the ocean. I'm too tired to talk about this right now. We can talk about it another time. But this is just an image from the Mediterranean Sea of radiance coming out of the ocean at 412 and 490, and they're on the same scale. And so this is the backscattered light, right? And so what is this going to look like? If this is 412 and this is 490, when you look at it with your eye, what do you see? Is this bigger or is this bigger? So it's very blue, right? It's very blue. And there's a lot of variability in the intensity of that very blue light. There's not a lot of variability in the intensity of this 490, right? So thinking about the spatial patterns here in terms of the backscattering of that light back up to the satellite caused by particles of different sizes and different compositions and et cetera, et cetera. So that's why we care about backscattering. And the Mediterranean. All right, lunchtime. Any questions later?
So again, 1:30?
Yes.
Today we're doing
The particle size what?
nutrient absorption.
Mm-hmm.
Scattering will be
Yeah.
more.
Yeah. Yes. So that, and also